Leah Lenzo, who is from the University of Nevada at Reno, and she will be presenting on seed mass response to weather variability for 4 Great Basin forbs and grasses.
Thanks, Gwen. Hi, everyone. I'm Leah Lenzo. I'm a PhD candidate at the University of Nevada, Reno in Dr. Beth Ledger's lab. I'll be talking about how seed mass responds to annual weather over 3 years in 4 Great Basin plants. Next slide. And we know that seed mass is a highly variable trait important for predicting plant performance. It's linked to germination, survival, establishment, dispersal. Next slide. But we don't know so much about how and why plants may adjust their seed size, especially over time. We know that parental size and resources play a role. Plants are under this trade-off between seed size and seed number with a strong pressure to produce more offspring. But we don't fully understand what conditions may lead to plants adjusting their strategy. Next slide. So we set out to ask how seed mass varies within the same population over time and how seed mass varies within the same maternal plant over time. Next slide. We selected 4 focal species native to the Great Basin. These are common species, widespread. They often co-occur together, which allows us to visit the same site and ask how these species may respond similarly or differently to the same stressors and cues. So we selected Indian ricegrass, arrowleaf balsamroot, squirreltail, and needle and threadgrass. Next slide. We selected our populations using historic seeds of success sites, all within the Reno area. And although some of these sites are close together, they are very different communities exhibiting very different pressures. So our Moon Rock site in purple is highly degraded, highly invaded. It's a popular recreational site. You can see all of the cheatgrass there. Our off Red Rock Road site is a super healthy forb-dominated site, really beautiful, a lot of Wyethia on that hillside. Our green southern site is up in the Pine Nuts Mountains, so it's cooler, wetter, more shaded. And then we had to add an additional Balsam oryza population at Sand Hills, which is a lot sandier and shrub dominated. Next slide. We followed modified collecting protocols, collecting all seed from 30 maternal plants per population per species for a total of 360 maternal plants each year. We tagged and GPSed each individual maternal plant so that we could return to it year after year. And we used a metal detector to relocate some of these tags. Next slide. After we collected the seed, we brought it back to the lab where they were cleaned and weighed individually by maternal line. That way we had 30 seeds from each maternal plant that we knew its identity, we knew its weight, its population, and year of collection. Next slide. So using all of this data, we can ask how does seed mass vary within the same population over time? Next slide. I won't show all of the environmental data here. We're still determining our covariates, but all 4 of our sites consistently over the last 3 years became cooler and wetter. And we would expect that with these seemingly more favorable conditions that our populations and maternal plants would produce bigger seeds from years 1 to 3. Next slide. But that's not exactly what we found. Here, each panel is an individual species where ricegrass is on top and then Balsam oryza, squirreltail, and stipa. Our columns are the sites. So Moon Rocks on the left was that highly degraded site. Pine Nuts is our high elevation site. Red Rock is our forb dominated site. And then Sand Hills is that extra Balsam oryza site. The box plots are colored by year, 2021, 2, and 3. And our y-axis is log transformed seed mass so that we can compare our different species together. And you'll notice that Balsam oryza seems to consistently produce larger seeds from year 1 to 3, seemingly preferring these cooler and wetter years. But our grasses like ricegrass and stipa either had a downward trend, particularly in pine nuts, or preferred intermediate sites. And the pine nut site likely is due to a reduced growing season. It was so cool and wet in year 3 that there was still snow well through May. Next slide. And we can also ask the same question at the maternal plant level. So here is an Indian ricegrass plant with 2 tags, meaning we found it in year 1, we found it in year 2, and here we are again in year 3 collecting that seed off the same plant. Next slide. And here, similar to the last figure, but this time the columns are species with ricegrass, balsamorrhiza, and stipa. Our colored rows are our sites. Apologies, the colors don't totally line up, but moon rocks, that degraded site is up top, followed by pine nuts, red rock, and sand hills. Here I've removed squirrel tail because we're still weighing those seeds, so we don't have full maternal data yet. The x-axis is years, so 2021, 2, and 3, and the y-axis again is log transformed seed mass. But this time each point is a maternal plant, so the mean seed mass produced by that maternal plant, and they are connected by lines. So the line connects the same maternal plant year after year. And you'll see balsamorrhiza typically had consistent response at the maternal plant level, where 2 of our sites produced bigger seeds in years 1, 2, and 3. But our additional Sand Hill site was very a varied response at the maternal plant level. And then our 2 grasses here also exhibit varied responses at the maternal plant level, either consistently decreasing in their seed mass, especially in our high elevation pine nut site, or just all over the place, like our Moon Rock site, where we have maternal plants increasing year after year, decreasing year after year, or showing some sort of intermediate change. Next slide. So just to conclude, we found that seed mass varied among nearby populations experiencing similar weather and climate, possibly due to either genetic factors, community structure, or competition. I didn't show it here, but I also gathered data on each maternal plant and herbivory. And interestingly, we found that balsamorrhiza tended to have a more consistent response in seed mass with cooling temperatures and increasing precipitation. Next slide. I want to give a quick shout out to the International Network for Seed-Based Restoration. We are a section under SER with a lot of great resources. Highly recommend checking this out. Next slide. And with that, thank you. Thank you in particular to the Bureau of Land Management, Plant Conservation and Restoration Program and the Nevada Native Plant Society for supporting this work. My contact information is below, and thank you.
Thank you, Leah, for that really cool presentation. I don't see any questions in the box right now, so it might be time to move on to the next speaker. Next up, we have Bill Davidson from the USGS Forest and Ecosystem Science Center, who will be talking about landscape and management of co-treatments influencing longer-term establishment patterns of post-fire aerial sagebrush. So you can go ahead and turn on your camera now.
All right, am I good to go?
Yep.
All right, great. First of all, thanks for the opportunity to present some preliminary findings from our research. I'd like to start by thanking our funders and collaborators, the BLM and JFSP, as well as the FIRES crew here at our lab that made all this work possible. Sorry about the long title. It's kind of a hobby of mine to make the longest titles possible. So I think I'm succeeding in that. So next. All right, so the question is, how do we reestablish sagebrush following wildfires, which has been historically and notoriously very difficult? So next. All right, so all of the data I'm presenting on today comes from the Soda wildfire that burned 113,000 hectares on the Idaho-Oregon border, seen there in panel A, and the Oahe Mountains, which is a very topographically heterogeneous landscape. The dots in panel B represent our monitoring plot distribution. Next. All right, so following the wildfire, the BLM implemented a number of management treatments seen in panel A, such as herbicide. These treatments were conducted over several years. So you see the solid blue color the first year, and then the hash lines for the second year herbicide treatments. Drill seedings of introduced and native perennial bunch grasses. See the sagebrush seeding itself. And then D, they also aerially broadcast some of those perennial grasses. Next. So especially with the topographical complexity of the study site, it allowed us to ask a number of questions. So what factors influence sagebrush seeding establishment? How do other land management treatments that overlap those treatments affect sagebrush? And how does first year emergence inform those longer term establishment patterns that we see? Next. So first of all, I just want to make a few points. It's really important to monitor first year after treatments. That'll really help learn and improve the effectiveness of these treatments. For instance, without monitoring in the first year, if you don't see any sagebrush establishment, you have no way of knowing whether it's issues with seed viability, for instance. Next slide. It's all sagebrush establishment is also can be very patchy across the landscape. So it's important to have a good density of monitoring plots so you don't miss those areas where you are actually seeing establishment. Next. So once you identify these sites, then you can ask, what is it about these specific sites that are conducive to sagebrush establishment? Next. All right, so we used a frequency density approach to quantify sagebrush presence and density. At each plot, we also characterize the vegetative community as well as a fertile island cover. Fertile islands are the legacy effects of burnt shrubs or perennial grasses, and that the combustion of those materials alters the biogeochemical properties of the soil. So they have increased nutrient concentration, for instance. We also characterize the pediderm, which characterizes that soil surface characteristics, excuse me. For instance, if the plot is dominated by exposed bedrock, you're probably not going to see any establishment there. Also, the soil redistribution class is important. For instance, sagebrush seed germinates best very near the soil surface. So if there's a lot of soil redistribution on the plot, then those seeds can end up quite deeper in the soil, which reduces their probability of emergence. Next. So we established over 2,000 monitoring plots in 2016, seen in the figure on the right, and monitored those in 2016 and then resampled those in 2020. We use generalized linear mixed models to assess the factors that influence the probability of a given plot being occupied by sagebrush. So it's more of a presence absence metric that I'll be presenting today. And so we use the mixed models to look at the plot level data that we collected, as well as the climate of the site, as well as the topography of the site. And then we used CART models, which is the classification regression trees, because these models really they generate decision trees essentially that are really a useful interpretation for managers to refer to when they're planning their management treatments. Next. So we previously published on the initial emergence results. And overall, we found that aerial seeding of sagebrush increased the probability of site occupancy by 12-fold. And we'll come back to this figure on the right, but I do want to point out that even in the lowest elevation, most heavily invaded sites, many of which have very alkaline soils, even in those harshest of sites, we saw a doubling of seedlings. But seedling mortality is typically highest in the first few years. So in order to characterize that long-term establishment requires later monitoring. Next. So from 2016 to 2020, we saw complete mortality on 17% of our plots. So I'll be presenting our results as things that promote sagebrush occupancy, but you could just as likely look at the inverse of that as sites where we observe mortality later on. Next. So when looking at these data, all these slides are going to be formatted similarly. So we have the 2016 results on the top, that's the emergence data, and then longer term establishment patterns on the bottom, all of which probability of sagebrush occupancy is on the Y and the factors on the X-axis. So we saw a promotion. Oh, sorry. Yeah, so we saw the highest probability. Sorry, can you go back? Yeah, so factors that influence those initial emergence patterns were consistent between 2016 and 2020. So that really tells us, again, that we're more in the 2020 data, we're looking at those areas that affected mortality patterns. So heat load, which is a measure of solar warming, topographical wetness index, and elevation all influence the initial emergence patterns, which were similar to 2020. However, instead of elevation, the mean temperature of the coldest month came out as significant for those longer patterns. Next. So in 2016, the pediderm was one of the most significant factors. So we saw the highest probability of occupancy on bare grain soils and soil aggregates as compared to the other pediderms. However, in 2020, rather than the pediderm being important, the deeper soil characteristics were more significant. So we saw the highest probability on mollisols, which have a higher content of organic matter and nutrients, as well as deeper soils that lack extensive restrictive horizons. Next. In 2016, the vegetative community played a significant role, especially the detrimental effect of exotic annual grass cover. Perennial bunch grass cover had a parabolic relationship. It was kind of a sweet spot, the middle values. And then we saw a really strong effect of fertile island cover promoting sagebrush emergence. We didn't see those same effects in 2020, but rather the co-treatments that weren't significant in 2016 did come out as important for longer term establishment. So you can see in the figure on the bottom right, with the management treatments on the X and the proportion of plots that were occupied on the Y. Next. So if you remember the initial emergence patterns, even though management co-treatments weren't significant, we did see a significant detrimental effect of exotic annual grass cover. And so it only makes sense that the herbicide application, for instance, that reduces that exotic annual grass cover that my coworker Kara has previously published would have an effect on that. Next. So when we compare, these are model predicted occupancy across the study area based on our data. So in both of these figures, the lighter the color, the higher the probability of sagebrush occupancy. So again, if we look at this as areas where there was mortality in subsequent years, you see the darkening in those regions. Next. And these are the CART models. So this is the initial CART model from 2016. Again, that just really presents the data in this decision tree format that is really, I think, intuitively useful for land managers. So factors that promote emergence was aerial seeding of sagebrush, saw higher occupancy at higher elevations, higher cover of fertile islands, and sites with lower heat load. Okay, one more slide. As compared to the 2020 results that really emphasize the soil taxonomic subgroups that are deeper and lack those restrictive subhorizons. And then the final slide. Oh, sorry. Conclusions, aerial sagebrush seeding resulted in a 60% increase in the probability of occupancy over unseeded areas. Land management co-treatments that reduce exotic annual grass cover, or the drill seeding of perennial grasses promoted sagebrush establishment. And again, the contrast between the pediderm, the soil surface being important versus the deeper soil characteristics. And then the thank you slide. Thanks.
Thank you so much, Bill, for that really interesting presentation. We have several questions, but we need to move on to the next speaker, so we'll just move these over to Huva.
Excellent. Thank you.
All right, so up next is Jesse Gray from the Aridlands Laboratory at the University of Colorado-Boulder. And they're going to be talking about approaches to revegetation following Pinyon-Juniper slash pile burning. So you can go ahead and turn your camera on.
Thank you. Good afternoon, everybody. Today I'm going to share an overview of the early results from a shrubland restoration experiment. I conducted this last year on land managed by the BLM in the Pinyon Basin, which is in western Colorado. Next slide, please. These ecosystems are irreplaceable for a variety of wildlife, including greater and Gunnison sage grouse, both of which are near endangered and in decline. These birds build their nests within sagebrush plants and really nowhere else. And unfortunately, sage grouse populations are seriously threatened by human activities involving habitat fragmentation, noise pollution, livestock grazing, and even just our presence. They're pretty shy birds. Next slide. However, my focus today is not on the usual suspects, but rather on the encroachment of pinyon and juniper trees into sagebrush ecosystems. This is a natural migration, partly due to climate change, but it poses a significant threat to the sage grouse. These birds avoid areas with trees, even former nesting sites, because they often harbor predators like red-tailed hawks that can easily prey on the female grouse. Next slide. To address this, the BLM commonly removes pinyon and juniper trees using chainsaws and then stacks them into slash piles for controlled burning later in winter. This is a method that's effective for habitat restoration, and at the same time, it causes minimal disturbance, at least on the landscape scale. However, while each pile is only around 6 to 12 meters in diameter, they burn much hotter than your typical wildfire, causing high disturbance on a very localized scale. And the number of piles required can also be significant, leading to considerable disturbance in aggregate in some of the projects. What our study aimed to do was test revegetation methods within these burn scars and hopefully develop a simple, quick, and effective approach to revegetation that field crews can use following those burns. At its core, this experiment was an evaluation of a custom seed pellet and whether it could surpass well-established but more labor-intensive restoration methods in its effectiveness in germinating native seeds. We used a diverse native seed mix collected from various Colorado climates with the additional aim of hijacking burn scars as nucleation sites for introducing future adapted plant genotypes and species. And finally, we wanted to determine how much of a problem herbivores might be for achieving success if our methods were ever to be implemented in real-world vegetation projects. Okay, next slide. We designed our seed pellet to address multiple anticipated germination barriers. It included an absorbent gel in the core for better water retention, a non-ionic surfactant to reduce soil surface hydrophobicity that's associated with high-intensity fires. And its structure was expected to deter seed predators compared to naked seeds, at least. It also included activated carbon to sequester organic compounds away from the seed-containing core so that we could apply a pre-emergent herbicide at the same time as seeding and reduce competition from non-native annuals, mainly cheatgrass and a lepidium species that I never fully identified. Next slide. The established restoration methods that we used to compare against our pellets included furrow raking, some light mulching to retain water and humidity and reduce the amount of wind throw. It gets pretty windy up there. And we inoculated the surfaces with nearby unburned soil to replace lost microorganism communities. And these modifications were all lumped together into the same treatment for comparison. In addition, the grazing exposures, which we see there in the photo, were placed on half of our plots, and a 2% solution of imazapyr was applied to the downhill half of every plot. Next slide. Before I start showing any bar plots, here is a photo of our most successful plot, with the top half showing very high germination from unpelleted seeds, and the lower half with the imazapyr showing none. In fact, across all 96 of our plots, only a handful of seedlings emerged in these herbicided halves, regardless of whether or not the seeds were in pellets. So we didn't end up gaining much information from that treatment, except maybe not to use 2% imazapyr for this. Next slide. So this is a distribution of the number of seedlings that germinated per plot, and sadly, most of our plots had no seedlings. I think this is likely due to a late seeding date after burning, which was delayed for several months. Additionally, while Central Colorado saw a nice wet growing season, our Western Colorado site faced unusually dry conditions last year on both the 50 and 10-year scales, the latter of which is considered part of a long-term drought. Next slide. Looking at the effects of the seed pellets, we were disappointed but not entirely surprised to find that they actually reduced the rate of germination. This may be related to the unexpectedly long waiting period before seeding. During that time, the pellets hardened quite a bit. It wasn't until after the late season monsoon rains that I found them to have really softened and broken down much at all. But in general, the practice of pelleting is still pretty much in its infancy in restoration compared to agriculture, where it's commonplace. So I still hold out hope for this technology in the future, but it's really tricky when the environment is so much less controlled than what you see in agriculture. Next slide. Okay, so next up is the combined effects of the established restoration tactics. Here we see a strong, though expected positive effect, indicating that the labor-intensive environmental modifications were comparatively worthy of the extra effort. And really all the additional equipment we really needed to do this was a rake to dig those furrows out. The mulch itself came from parts of the trees that hadn't fully burned, and the inoculant came from just soils that were a few feet away. Next slide. Skip this slide, please. Okay, so during my later visits, I did find some visual signs of nibbling on some of the seeds or seedlings that were in plots without exposures. And in this figure, we see a weak indication of grazer impact, but with all the overlap and the variability here, it's not really clear that herbivores were a major factor in our results. Next slide. Okay, however, there was a pretty clear effect of ambient environmental conditions. Prior to burning and seeding, we had measured several potentially influential factors, including hill slope and aspect, soil acidity and moisture content, the size of the slash piles, and soil texture. And the last of these does seem to be important. I analyzed seedling emergent rates by soil texture, comparing actual versus the expected numbers we would see if texture had no influence. With more sandy loam plots than any other texture, for example, more seedlings were expected in the sandy loam. And we see that in the dash bars here. So those are the estimated number of seedlings in the sandy loam. However, while the actual germination rates in sandy loam exceeded expectations, the others either matched or underperformed, as indicated by the accompanying solid bars. While it would have been better if we had equal representation among these textures, that wasn't ever a part of our original experimental design. And still, it does suggest that we might benefit from seeking out these types of soils, the sandy loams, when deciding where to place the slash piles in the first place. Next slide. I will be visiting the site again in the spring to see if there are any additional germinations from the pelleted seeds. The naked seeds have all been blown away or been eaten, but the pellets are still there. So I'm hoping that more time under the snow will give them a second chance. And with that, I'll leave the summary up here, and I'd be happy to take any questions or suggestions. Thanks.
Thank you, Edward. Okay, I'm seeing a few questions over in the Q&A. One is, why did you choose imazapyr for your herbicide treatment?
So imazapyr is used as a pre-emergent commonly. And what I had hoped is that it, even though I expected it to affect the naked seeds, what I had hoped is that the activated carbon within the core of the pellets would prevent the herbicide from reaching the seeds that are in those cores. And so those would be able to germinate, while anything else, including the naked seeds and including any cheatgrass in the seed bank, would be affected by the imazapyr. Going forward, I don't think I would use imazapyr. I think it's more effective than I needed. I would maybe go for something like indaziflam, or just skip it altogether and hope for the best.
Thank you. There are a number of other questions over here for you, but we need to move on to the next speaker, so these will be moved to Huva.
Thanks.
Great. So up next, Catherine Silliman from the University of Nevada, Reno, will be talking about seed emergence, timing, plant size, and seedling survival of winterfat. So you can go ahead and unmute yourself and turn on your video.
Awesome. Hi, everyone. My name is Kathy Silliman. I'm a master's student at the University of Nevada in Reno, and I'm excited to share my work with the shrub Krasceninnikovia lanata, also known as winterfat. So let's jump to the next slide. To paint the scene, here's one interpretation of the Great Basin and the Mojave deserts, which make up a large area of relatively undeveloped land in the United States. It is home to a wide distribution of plants and animals and allows researchers to study how different species vary across this wide range. Next. However, it is not a perfect system. Both deserts are experiencing dramatic changes in the form of wildfires, weed invasion, and climate change. In respect to climate change, it will likely increase aridity, and if changes are faster than seedlings are able to evolve, then maintaining native plant populations might require the help of humans. Next. And humans are already helping with the use of empirical and provisional seed zones, but this method may be challenging if seeds are unable to germinate and persist. So choosing the right seed source for successful restoration might rely on populations best suited for future climates. So one way to find this out is just to move seeds around and see what happens, which is what my project aims to do. I'm investigating the movement of populations from their drier southern range to their northern range in order to match potential future climates. Next. Let me introduce you to the superstar of this talk, Krasceninnikovia lanata. It is a perennial shrub that can be found in much of the western United States. It has a high forage quality that is important to many wildlife, hence its common name, winterfat. Next slide. So here's a map of where I have seed collections and my common garden sites. The dark blue circles are collections that I made, and the light blue squares are collections from the Seeds of Success that I received from the Germplasm Resource Information Network. And then the yellow triangles are my field common garden sites where where I'm testing the movement of populations south to north. Next slide. So with those range of collections, I had a couple questions I wanted to ask. One, are there differences in seed emergence, seedling survival, and seedling size among populations when grown in different settings? And if so, does the variation of the collection site help explain those differences? Next. To answer those questions, I performed these 3 experiments. Krilla A, where I took seeds and I grew them in the greenhouse or in a greenhouse common garden. Krilla B is where I took those same seedlings from Krilla A and transplanted them into 3 field common garden sites. And Krilla C, where I sowed seeds directly into the field. Next slide. For the first experiment, Krilla A, I chose 15 populations and I planted 98 seeds per population, so one seed in one tube. And then after a month and a half, If the seedling either died or the seed never emerged, I replanted that tube with the same population. So this experiment has 2 rounds of planting. Next. For both rounds, I recorded emergence timing and survival. And then 4 to 5 months after planting, I recorded size and volume of the above ground biomass. And then these seedlings stayed in the greenhouse for about 9 months until I was ready to transplant them into the next experiment, which is on the next slide. So I assigned a third of the seedlings from each population to one of the 3 gardens, and then I hardened them off before transplanting. Next slide. And then after being hardened off, the plants were transplanted into their respective gardens. Next slide. And so this is what the Gerlach garden looked like and my Fields garden looked like. Next slide. And then the last third that were to go into Reno, I uppotted because the soil type at our greenhouse complex just had too much clay. And so this was also guaranteeing me that I had some data in case all of the plants in the field actually died. And then throughout the summer, I recorded survival and size. Next slide. And on to my last experiment, C. I sowed 25 seeds per population at the Gerlach garden, and I recorded emergence, survival, and size throughout the summer. Next slide. Let's take a look back at my questions, which were, are there any differences among populations when grown in different settings? And if so, does the variation of the collection site help explain those differences? Next slide. For all my analyses, I ran generalized linear models with and used spatial variables and environmental variables as needed. So my spatial variables were latitude, longitude, and elevation. And then I used the 7 environmental variables based on Alison Honoré's work that she did in the Great Basin, where she found that these 7 were important predictors of seed emergence in other species. Next slide. And so for the first experiment, A, I found that populations did differ in their emergence timing. So this graph is showing the mean hours to emerge for each population, with the fastest taking about 100 hours on average to emerge, so about 4 days to emerge on average, and the slowest taking about 6 days on average to emerge. And I don't want to bore everyone with too many graphs, so I'll just say that populations from more western, wetter, lower elevation, and more seasonal locations had faster emergence. Next slide. And then after growing the greenhouse for about 4 to 5 months, I found that populations also differed in their size. So this graph is showing the mean size and volume for each population. And after running some more fun GLMs, I found that populations from more southern, lower elevation, and more seasonal locations tended to be larger. Next slide. And then onto analyzing the field experiments for B. I want to preface that populations still differed in their size before transplanting. So this is a jitter plot showing that there was size variation between populations, but also within populations. And this graph is also showing the number of individual seedlings in each population that ended up being transplanted. Next slide. I also want to say that I analyzed the Fields and the Gerlach's garden separately than Reno because the conditions were so different. Reno plants were babied and watered, whereas the Gerlach and Fields garden had to fend for themselves. And so after the summer, I recorded survival at all 3 gardens. But here is a graph showing percent for survival for the Fields and the Gerlach garden. I want to point out one outlier for Grin 1. There was 3 plants grown at the Fields garden and all 3 survived, whereas the 2 planted at Gerlach both died. But you can tell from, you can see from the other populations though, there are differences in survival. It also looks like the Gerlach garden has slightly higher survival than the Fields garden. However, the effect of population and the effect of garden and the interaction between the 2 were not significant in predicting survival. Next slide. The only variable that I found to predict survival in the field was the initial planting size of the seedling. So here's a jitter plot that shows the individual plants and their initial planting size and whether that plant survived the summer or not. So as you can see, there are variation in the initial planting size. However, there was a significant effect of the initial planting size on survival. So larger plants tended to have higher survival. Next slide. And the same can be said about the Reno garden, which had higher survival, but survival was predicted only by the initial planting size. Next slide. And lastly, analyzing the data for my last experiment, Coryllis C. This graph is showing the difference in seed emergence for each population, and populations did differ. However, I couldn't find any spatial or environmental variation that could help explain that. So something else is going on. I just don't know what at this point. And then the 3 stars represent the 3 plants still alive at the end of summer out of the 111 seeds that emerged. Next slide. And so the story so far is that populations from more western, wetter, lower elevation, and higher seasonal locations had faster emergence, and plants that were larger were found from more southern, lower elevation, and higher seasonal locations, and larger plants tended to survive better when transplanted into the field. And under field conditions, populations did differ in their emergence, but no climate or spatial variables that I've looked at could help explain that, and survival was very low. The next steps for me are to continue monitoring these gardens throughout the winter and see if these southern populations can handle the cold, because if these southern populations tend to be larger and larger plants tend to survive better and they can handle the cold, then that will be useful information for land managers and future restoration projects. And so lastly, on the last slide, there are quite a few people that have helped me along the way that I would like to thank. But with that, I'll take any questions if we have time.
Thank you so much, Kathy. That was really interesting. We do have to move on to the next speaker, but there are questions for you that we'll just move over to Huva. So next up, Michaela Owens is a native plant scientist at the Nature Conservancy in Wyoming, and they will talk about efforts to improve Artemisia triedentata establishment with seed technologies. So go ahead and turn on your video and mic.
Great. Yeah, thank you all so much. I'm happy to be here. My name is Michaela Owens. And yeah, I'm going to talk to you all today about our process and the challenges we've faced trying to find an effective seed technology for enhancement for Artemisia triedentata. And I also just want to say we've had a lot of great sagebrush presentations today, so I'm really excited to kind of add to all that we've heard so far. Next slide. So the sagebrush steppe, as you all may know, and many of you seem to work in, is a widespread and iconic landscape of the American West. And while it used to be as large as 500,000 square miles, today we've lost as much as half of that. And unfortunately, the loss continues with around 1 million acres being lost every year to invasive species, wildfire, climate change, and development. But we're lucky in Wyoming. This is where we have the largest tracts of remaining high quality intact ecosystems. Next slide. So yeah, Wyoming big sagebrush is a foundational species in many of the sagebrush ecosystems within the sagebrush steppe. And within the sagebrush steppe, there are around 350 species of concern. So that makes this landscape of special importance to conservation. And within this landscape, we do have some species that can only be found in the sagebrush steppe. Probably our most famous endemic species is the greater sage grouse, pictured in the upper left-hand corner of this slide. But it's also home to a number of other amazing species, such as mule deer, pronghorn antelope, pygmy rabbits, and burrowing owls. And all of these species rely on sagebrush at some point in their life cycle. So it's important that we restore these species, when we can. Next slide, please. So as the sagebrush steppe continues to be threatened, the need for restoration is only going to increase and become more important. But unfortunately, there are a lot of challenges with establishing sagebrush in reclamation and restoration. Transplanting is pretty successful usually, but it is prohibitively expensive and time-consuming, especially on large scales. So for a lot of restoration projects, this means the only viable option is seeding. But there is risk in seeding, as seeding failure is often what comes out of seeding sagebrush. So you frequently get landscapes like this one on the right, where you get a lot of perennial grasses that reestablish, but little to no sagebrush. And there are a few different reasons for this. Typically, sagebrush in a natural setting establishes episodically. So during infrequent good years with lots of spring moisture is when you see sagebrush come in high numbers. And those sorts of years are becoming less frequent because of climate change. Water and nutrient availability is also usually an issue for sagebrush during that first growing season. Competition can also be a problem, especially from invasive annual grasses, but also if there's established perennial grasses or other species on a site you're trying to seed into, that can also be a barrier to establishment. In certain restoration settings, and especially like in mine reclamation, where we do a lot of work, soil stability and erosion can be a problem because the seeds can get buried if there's a lot of soil movement, which inhibits germination. And lastly, seed viability can be a big problem because sagebrush seed only stays viable in the seed bank for up to a year. So if you don't get good establishment after that first seeding, it's not going to come in later on. Next slide. So the approach that we're exploring to overcome these challenges is seed technologies, and seed technology is a pretty widespread approach used in agriculture where seeds are treated or packaged in active ingredients that modify their germination or growth in some way. And so the seed technology we're trying to develop for sagebrush is root enhancement seed technology, which, as the name implies, we hope this seed technology would enhance root growth for the first growing season, which would give seedlings better access to resources deeper within the soil profile. And then we think this will improve establishment, survival, and drought tolerance. And our end goal is to identify a seed technology that accomplishes these things without impacting emergence, and that is scalable for use in restoration and reclamation. Next slide. So to try to develop this type of seed technology, our first step was to look into fertilizers that were already available on the market that we hope would provide root enhancement effects. And so we started by testing a few in our seed lab. And so to test these, we use these PVC pipes that we have cut in half and then tape them back together. We put weed cloth on the bottom and fill them with a 50-50 mix of local soil and sand. And then we sow each pipe with a number of seeds, thin it down to 1 seedling per pipe, and then we grow each seedling for 40 days from emergence. And then because of the pipes being cut in half, we're able to open those up and take a look at the root growth. So we look at root length, below ground biomass, and we also look at above ground biomass, shoot length, and emergence. And we compare between the pipes that were treated with a fertilizer and bare seed. Next slide, please. So of the fertilizers we tested initially, there was one that showed good promise. It's called Root and Grow. It's a liquid fertilizer manufactured by Bonide, and it's really high in phosphorus. And so this fertilizer provided the desired effect and produced seedlings with significantly greater below ground biomass as well as root length, as you can see in this plot here. Next slide, please. So once we had identified this fertilizer that we found to work well, we had to find a way to deliver this fertilizer to the seed, which has been the challenging part. We initially started out by creating our own proprietary seed pellets. So we took the fertilizer, some binding ingredients, and bare seed and mixed it all together and made these extruded pellets you can see here with a pasta maker at our seed lab in Lander, Wyoming. Next slide. So we tried several different pellet recipes and determined pretty quickly that pellets were just not going to be a viable vehicle for nutrient delivery as they inhibit germination, as you can see here in this plot. Next slide, please. So because the pellets were not going to work, we have moved on to trying other types of seed technologies. We formed a partnership with a seed technology company. And because of the negative impacts to emergence inherent with coating sagebrush, we focused our efforts on film coatings that have already been developed for agricultural settings, because unfortunately we weren't able to turn the fertilizer we knew worked into a film coating. So we've been working with things that our partner company has already developed. So this slide here just shows some bare seed and some seed coated in a film coating, which is just like a light coating on the seed. Next slide, please. So sure enough, we did find that the film coatings did not inhibit emergence, which was great. This box plot here, I think, is from our 10th trial. We've done a lot of different iterations within this work. So that's great news that the film coatings don't inhibit emergence. Next slide. But unfortunately, we just haven't found that there are any effects on root growth from the film coatings. We've run a number of different trials with our PVC pipes in the lab with different film coatings where we are able to directly observe how the roots are being affected, the root growth is being affected, and we've also tested these out in the field at reclaimed mine sites. And in both settings, we just haven't found any evidence of any effect to root growth or survival from any of these film coatings. So at this point, we've come to the conclusion that film coatings are also not a viable way to enhance root growth because you just can't get enough of the material onto the seed without inhibiting emergence. Next slide, please. So that takes me to our last and latest idea. We have been working on a new seed technology that we're calling seedi. They're named after a tasty South Indian snack that looks just like these kind of seed balls that we've developed. So our hope with this is that we can deliver the fertilizer to the seeds without impacting emergence. So basically what these are in the top left corner is a picture of the seedi. They're nutrient balls, kind of in a throwback to our original pellet idea. It's got binding ingredients and that root and grow fertilizer in a ball, and then the seeds are encrusted on the outside. So hopefully the seeds slough off as the ball dissolves over time and they're able to germinate. And these are also great because we do get to use the fertilizer that we know works, and we get to use it at a concentration that we know works. So we're really optimistic about these. We have done a couple of small lab trials so far. We currently right now have one of our PVC trials underway, and we have put these out in the field at a reclaimed mine site. And so we'll be gathering data on whether or not these work over the summer, and hopefully we'll see some good results and are able to move on to testing them at a larger scale at some point in the future. Next slide. So with that, I just want to say thank you. Thank you to my collaborators. Thank you for having me. And thank you to our funders. And if there's any time for questions, I can answer them.
Thank you so much, Michaela. I'm excited to learn about how the trials with seedi go. We don't have time for questions right now. We need to move on to a break, but we'll move all the questions over to Huva for you to answer. Thank you again, and we'll see everybody back here at 1:10. Oh, 2:10. Different time zone. Thanks, everyone.